guys, today's video is about the animal cell and the plant cell. So to save time, I have drawn the diagrams, but I will explain them in detail. So before we start, a quick recap. Organisms are divided into two parts, eukaryotes and prokaryotes. So eukaryotic organisms are the organisms that have an organized nucleus, whereas prokaryotes don't have an organized nucleus. And the other difference is that eukaryotes have other membrane-bound organelles such as the mitochondria. Prokaryotes, they don't have other membrane-bound organelles as well. So going to the examples, eukaryotic examples are the animal cell, plant cell, fungi, protozoa, whereas the prokaryotic example is the one and only bacteria. So in today's video, we will be focusing mainly on the animal cell and the plant cell, whereas we will be discussing fungi, protozoa, bacteria and viruses in another video. So we'll start off with the animal cell. So I've drawn a diagram of the animal cell right here. An animal cell does not have many, this is called a typical animal cell because we have included almost all the possible organelles into one cell. This is not how all cells are going to look like, but a typical diagram means it contains all the possible organelles. So we'll start off with the cytoplasm. We'll start off with the cell membrane to make things easier. The cell membrane is the outermost layer in an animal cell. So it is semi-permeable. You have to remember that a cell membrane is semi-permeable. So this guy right here. It only allows certain substances to enter and leave. So in my terms, it acts like a security or a police officer that controls whoever enters the cell and leaves the cell. It is found in both animal cells, plant cells, fungal cells, etc. But in animal cells, it is the outermost layer. That's a very important point. So from the cell membrane, let's go to the next organelle, the cytoplasm, the yellow color organelle that I have drawn there. So I'd like to tell my students to think of the cytoplasm as a ball of jelly. Get that in right. A ball of jelly, which the jelly is the cytoplasm. So this is what represents the cytoplasm. So in the cytoplasm, there are the organelles that are on top of it, like the decorations that are added on top of the jelly. So the cytoplasm is a slopy jelly-like structure that contains all the organelles, and it allows all the reactions that go on in the cell to take place. That means all the reactions that happen in a cell take place in the cytoplasm. Next organelle. I've forgotten to label it, so actually didn't label it. So this center organelle right here in the middle, that is called the nucleus. Now if we zoom the nucleus out, just draw it below this. If we zoom out the nucleus, it's a double membrane bound structure. It's not very clear in the first picture that we have drawn because it's a very tiny organelle. And inside this double membrane, we have the most important genetic material. So this contains the genetic material. So in case of an animal cell, our genetic material is, what is the genetic material? Guessed it? DNA. So DNA is a double helix structure. So for those of you who did not know that, it's something that looks like that. And this contains special structures called genes. We'll talk about that in a later video. These genes code for proteins. That means that determines what proteins are produced in a person's body. So the nucleus in return is the control center which controls all the reactions that go on in a cell. The next organelle. take you back to the diagram. If you can see, the mitochondria are labeled right there. I'm going to take the mitochondria out as well for a better, clearer explanation. Mitochondria, just like the nucleus, is a double membrane bound organelle, but the mitochondrial inner membrane is highly folded. So looks a little ugly, so I'll just draw one nicely. This highly folded inner membrane allows the mitochondria to have a high surface area and allows the reactions to take place in the mitochondria. So what are the reactions that take place in the mitochondria? We know the mitochondria as the powerhouse of the cell, providing energy. So in correct terms, it carries out the reactions of respiration. That means we have glucose reacting with oxygen to give out carbon dioxide, plus, this is a very messed up equation. I've not written it correctly. So just to make it accurate, I'm going to replace the glucose by its equation, C6H12O6, which also means I have to balance the equation. 6 CO2, 6H2O plus energy. So the most important part here is the energy that is given out. The energy that is given out is in the form of something called ATP. This is also known as adenosine triphosphate. Alright, so that is the energy that is produced in the mitochondria. So the mitochondria is the powerhouse of the cell. So a fun tip is that the mitochondrial membrane, a higher amount of mitochondria is found in cells that require a higher amount of energy. For example, like the muscle cells. The muscle cells require a higher amount of energy because they need more energy because they carry out more activities compared to the muscles in your ear cells. The muscles in your legs and the hands require more energy. So that is the mitochondria. So the next organelle that we will be discussing is the ribosomes. So below here I have typed all the meanings of the words. So once we finish explaining about each and every one separately, I will show you that. I'll show you the correct definitions. In case you want to take down notes, you can always pause the video and take down the notes. So we'll move on to the ribosomes. So the ribosomes are structures that look like that. They have two subunits. The small subunit, which is on the top, this is the small subunit, small guy, and this is the large subunit. It looks like large ribosome and small ribosome, so I'll just erase ribosome. But remember, this is the ribosome that contains a large subunit as well as a small subunit. So the ribosome has a very important function, that is, it carries out protein synthesis. Now I'll take you to the diagram and take a look at this rough endoplasmic reticulum that is labeled RER, the rough endoplasmic reticulum. So in the RER, there are attached ribosomes as well as the ribosomes that are freely moving. So there are two types of ribosomes in a cell, the ribosomes that are freely moving and the ribosomes that are attached to the rough endoplasmic reticulum. So this also covers the function of the rough endoplasmic reticulum. The proteins that are produced or synthesized by the ribosomes are transported and folded in the rough endoplasmic reticulum. Folding and transportation of the proteins produced by the ribosomes are carried out by the rough endoplasmic reticulum. There's another structure over here. This guy, the one that does not have any dots on it, that one is smooth. So it does not have a structure, it's just very smooth like that. It does not have the dots, no dots on it. It's very smooth. The smooth endoplasmic reticulum is different from the rough endoplasmic reticulum, and the SER has functions such as lipid synthesis. So that is where the lipids are produced. The Golgi apparatus also has another very important function. The proteins that are folded in the RER are going to be packed into vesicles, and they're going to be transported to the Golgi apparatus where they are modified. Modification of proteins take place in the Golgi apparatus. So once the proteins are modified, a modification, for example, it could be something like a lipid modification, where the protein gets a lipid added into it, so we can call it a lipoprotein. And then if we have such as the formation of a lipoprotein, so first I'll draw a Golgi apparatus. This looks very similar to the smooth endoplasmic reticulum, so it's very difficult to distinguish between the two of them under a light microscope. So inside the Golgi apparatus, these vesicles are the ones that bring the proteins that are supposed to be modified. So these vesicles come from the rough endoplasmic reticulum. They enter a modification. An example of a modification is when a lipid portion gets added to the protein. These are just random molecules, so this looks like glucose, so I'll just draw a different shape. That's much better. And this gets attached to it, so we get a lipoprotein. So this is one type of modification. Another type of modification could be when a glucose molecule is added to a protein, So when a glucose is added to a protein, that forms a glycoprotein. When these two combine together, that forms a glycoprotein. So these are the kinds of modifications that take place in the Golgi apparatus. So what happens once these modifications are finished? The same kind of reaction happens. These modified lipids or lipoproteins or the glycoproteins are going to be packed into secretory vesicles. These secretory vesicles then move towards the cell membrane and are sent out of the cell by a process known as exocytosis. Exocytosis is a process by which substances are sent out of the cell. So these proteins are sent to the place where they are necessary. So these are the functions of the animal cell. So here below I have written about each and every function. So we will go to this part once we have finished the plant cell as well. Now let's take a look at the plant cell, which is also a eukaryotic cell. So let's move on to the plant cell. So plant cell, I have drawn the plant cell as well. So if you take a look at the plant cell, you will notice that it has a little bit of different structures compared to an animal cell. The differences, I will highlight the differences. If you can notice by the time I'm highlighting them. So cell wall or vacuole, actually a permanent vacuole, and chloroplast. So in this, I will only be discussing about these three structures. So a cell wall, plants have a very rigid structure. That means they're strong. If you hit a plant cell, it's not going to break into two unless it's a very small stem or something. So usually big plants are really strong. That is because they have a cell wall. So their cell wall is made out of a very tough polysaccharide known as cellulose. So the cellulose cell wall has another important function. It is freely permeable. The cell wall allows any substance to come in and go out, so it does not care who comes and goes. Its only job is to give the plant cell a supportive structure. So it gives the plant cell a very rigid structure. Feature number 2, chloroplast. Chloroplast has a very similar structure to that of a mitochondria, which is why I have drawn them in two different colors. So I was going to write freely permeable here. So you know that now. So the chloroplast, just to make things understandable, I will use the color green. Chloroplasts are also double membrane bound. They have a very important compound called chlorophyll which gives them the green color. So the chlorophyll traps sunlight so that plants can carry out photosynthesis, the process which plants make food. So photosynthesis is the process by which they capture sunlight and convert carbon dioxide and water into glucose. I'm doing that again, C6H12, glucose and oxygen. So to balance the equation, so this requires two things. This requires chlorophyll. This requires chlorophyll as well as sunlight. So I'll just write the chlorophyll here. It just keeps going. Yeah, chlorophyll and sunlight. So in the presence of chlorophyll and sunlight, they can carry out a process called photosynthesis. So the oxygen that is actually produced after photosynthesis is the oxygen that we use for respiration. So that is feature number 2. So feature number 3 is a permanent vacuole. The reason why I use the word permanent vacuole is because we can have a vacuole as well, but not all the time. Plants do have a permanent vacuole. It's a single membrane bound structure which is filled with water and other inorganic mineral ions. So it maintains all the conditions or the necessary nutrients by the plant cell. So that concludes our lesson, but before I stop, I will rush through. The cytoplasm is a slopy jelly-like structure that contains all the cell's organelles, and yeah, I've mentioned it here as well, to think of it as a ball of jelly, as I mentioned in the beginning of the video. So it's a site where all the reactions take place. The nucleus is the control freak, which contains it's a double membrane bound structure which contains the genetic material of the cell, and it controls all the reactions that go on in a cell. The mitochondria, powerhouse of the cell, it carries out all the reactions of respiration in the cell to give out energy. Ribosomes, the site of protein synthesis, and RER, it has attached ribosomes, as the ribosomes can be attached to the RER or they can be freely found in the environment of the cell, inside the cell. So the ones that are attached to the ribosomes, they also carry out protein synthesis, and the products are transported and folded by the RER. Lipid synthesis, so maybe lipid synthesis takes place in the SER. We know the differences between RER and SER. RER has the attached ribosomes. SER is extremely smooth, as the name suggests, smooth endoplasmic reticulum. Golgi Apparatus is the editing unit where the proteins that enter from the RER are modified, and we spoke about how they enter through vesicles. And once they enter through the vesicles and the modification takes place, I gave you 2 examples of modifications, lipoproteins and glycoproteins, and they are packed into secretory vesicles and sent out of the cell by a process known as exocytosis. And we are here to the 3 features that are found only in a plant cell. So in case there's a question, if you have the doubt between the organelles that are present in an animal cell and a plant cell, voila, these are the organelles that are only found in a plant cell. So the difference between animal cells and plant cells is that a plant cell has a permanent vacuole, they have chloroplast, as well as they have a cell wall. So I hope you enjoyed this video, so give it a thumbs up and subscribe. So in the next video, we will be talking about enzymes.